So Dr. Smith today is going to be talking about the genetics of Dravet syndrome some more. I know Dr. Perry touched on that, but we're going to get a little more in depth. So just an introduction to who Dr. Smith is. He received his Bachelor of Science from the College of New Jersey, and then his medical degree from New Jersey Medical School. He completed his pediatric neurology and pediatric epilepsy training at the Children's Hospital of Philadelphia. And then he's been with the Minnesota Epilepsy Group for the last 4 years, where he started a clinic dedicated to the diagnosis and treatment of rare genetic causes of epilepsy, including Dravet syndrome. He brought the drug trial for fenfluramine for Dravet syndrome to the Minnesota Epilepsy Group and served as the principal investigator for the study site. Today he's going to present on what we know and what we don't know about the genetics of Dravet syndrome and try to help us understand what possible genetic treatments might be available in the future. Thank you so much for being here, Dr. Smith, and I'll turn it over to you.
All right, so let me first confirm you can see my slides, right? All right. All right, perfect. So thanks for the invitation. I really appreciate it. I love working with the Dravet community. You guys have an almost unmatched passion and really kind of helps on the days where I'm feeling less than passionate, keep me in check. And you're one of the inspirations why I wound up doing what I'm doing now in genetic epilepsy. Veronica kind of covered my credentials. I'm not a geneticist like Dr. Perry. I'm a neurologist. I'm just a pediatric epileptologist. And I kind of approach this from the perspective of what would I want to know if I'm in your shoes. I can go kind of in depth about the genetics, but at the end of the day, I wanted something that was useful for you guys. And so I subtitled it, It's Not That Simple, because I think for many of you guys, you know the simple story. And I realized as I was giving this lecture, a practice version of it, I had said it's not that simple about a half dozen times. And so I feel like that's kind of the summary at this point. What we're discovering now is kind of all the nuances and how things aren't so simple. And so without any further ado, let's dive right on into this. And obviously I have to start by saying that I have no financial disclosures to disclose. And so again, in asking myself, if I was in your shoes, what would I want to know? It's really primarily those last two bullet points, particularly what is this genetics going to mean for my child? Can this actually make a difference in terms of what treatments are available? And in order to understand those treatments and the diagnostics, the diagnostics are going to be bullet point number 3 and the treatments are number 4, I kind of need to get into the basics, which is first and foremost, what am I talking about when I say genetics? I think there's a lot of presupposition about what we know and what we don't know. And so I can't quite do genetics 101 in 30 minutes. So I'll just do genetics 100.1. We'll make this as basic as I can make it to understand exactly the concepts that we're talking about. Dr. Perry kind of stole some of my thunder there by talking about the diagnostics there, what we know about the genetics. I'll spend a little bit more time with it. And that's really where I start getting passionate. And this is really going to highlight what we know and what we don't know. And we'll talk a little bit about the practicality of genetic testing, and then we'll talk about the potential future of, and actually some of the present actually, of gene therapy. And so again, this is genetics 100.1. We got to make it as simple as possible. And so we call this the central dogma of biology. So it used to be, we thought this was a unidirectional process. We now have added all these extra arrows and realizing it's not that simple. But the important concept is that the way that the whole process of making your cells the way that they are is that the information is encoded within DNA. DNA is this double helix that you can see here, full of these series of letters that we call nucleotides. And they're essentially a series of 4 letters, T, A, C, G, or G, A, T, C, doesn't matter with the sequence of what you call them. And they're encoded in complementary bases that they kind of wind around each other in this double helix. Every single cell in your body has a full copy of all of the DNA. And each individual cell then chooses which portions of this to turn into RNA in a process called transcription. And so every cell has every bit of information in DNA. This is what's passed on to your children, but each individual cell will choose which ones it turns into RNA. This RNA serves as a single-stranded messenger. It doesn't have the both strands that we see in DNA. And this RNA, through a process called translation, is then turned into protein. And these proteins, at the end of the day, are what serve all the functions in your cell. They're the ones that make more DNA, that make more RNA, that make the proteins, the ones that make your neurons look like neurons, the ones that make your muscle cells look like muscle cells. We're still kind of understanding the process by which each cell chooses which portions to turn into RNA, and we're not going to cover that part in this talk. As I had mentioned, the important piece of this is that this DNA is what gets passed on to your children. And ultimately, it's not a perfect process. The fidelity is not perfect. There's on average about 6 typos per reproduction every time you have children. Now, there is a lot of DNA in your body. If I remember correctly, it's a bit somewhere in the odds around 20 billion of those letters, those nucleotides. Huge, 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 huge number. The vast majority of them, though, never get turned into RNA in any cell in your body. 99% of it is never turned into RNA. The 1% that is turned into RNA, which is then subsequently turned into protein, are what we call genes. These are the ones that actually get transcribed and actually serve the function of making these proteins. We're still not entirely sure what that other 99% does. Back when I was in high school, this is all junk. We don't really know what it's doing. Currently, we're starting to think that actually it is playing a pretty significant role, particularly in deciding which cells turn on which of those genes, which cells are turning those portions into RNA. And so they play a key role in the regulation of those genes. But that's still something that's actually quite, at this point, in its infancy. And then there's another layer of complication, because this isn't complicated enough. There's another layer of complexity in that there are ways in which your body modifies these genes that doesn't actually change the letters, and they kind of add little notes. Essentially, if you think of these letters as kind of like a book, just a series of these 4 letters, these are kind of little notes that your body writes in the margins. And we call that epigenetics, which we think are also now, we're starting only now to understand the role of epigenetics. And you're probably, since again, I know you guys are a very scientifically literate bunch, I'm sure you're seeing that term come up. Essentially, that's what epigenetics means, anything outside of the sequence of letters. But before we knew what DNA was, scientists knew what genes were because we understood how they worked. And the guy who first figured this out was a monk in the 1840s named Gregor Mendel. And now we're probably going back to high school biology for most of you guys. Mendel was a very, very, very meticulous man. And what he had noticed, and he was a botanist, he was actually interested in breeding pea plants. And what he had noticed is that there were 7 attributes, and so they're complementary attributes. So one attribute would be either green or yellow peas. It would be short or tall plants. It would be purple or pink, I can't remember which color for the flowers, round versus wrinkled peas. And he had noticed that for these attributes that they bred true. Every single time a green pea plant was mated with a green pea plant, it always made green peas. Same thing with the yellow, same thing with tall plants, same thing with short plants. But he had so he then asked himself, okay, what if we have these complementary or contradictory attributes? You can't have both a green and a yellow pea plant. Would you wind up getting some yellowish green color if you mate them together? No, that's not actually the case. What he discovered is that when you mate one of these true breeding green with a true breeding yellow, every single time, all of the offspring wound up having yellow peas. And he called that a dominant trait, that yellow color dominates the green, and the green is completely wiped out. And so he called then that green feature recessive. However, he discovered that the green didn't go away. It's still actually there and it's hidden. And it could essentially skip a generation, as we often talk about colloquially. So if you took these hybrid plants, the ones that were the daughters and sons of the green, of the plants that had a green parent and a yellow parent, they come together, they're all yellow. But if you then mate those together, that 3 out of every 4 will still have yellow peas, but 1 out of those 4 will be green. And he repeated this process for all 7 of those plants. So he determined that these attributes are passed down to their children, that some can dominate others. And based on that math, he had concluded that this is passed on in a way that you have 2 copies. And those 2 copies, again, you need, if you have 1 copy, it'll dominate the other one, but you need 2 of those quiet ones in order to overcome the dominant feature. And so pictured in our old friend, the Punnett square, what he had done, again, if you had taken a purebred yellow, so that would be big Y, big Y, big Y, big Y. Purebred yellow and a purebred green, you'd have big Y, and then little y up here. And in each case, you'd have big Y, little y in each case, and the big Y dominates the little y. And in each case there, you would wind up with a yellow seeded plant or yellow peed plant. But if you take their offspring, which would be all big Y, little y, so one of these guys in the upper left corner here, and you mate them together, what you wind up having are 4 possible outcomes. They can pass on either the yellow or the green gene, so the big Y or the little y. And if you wind up with big Y, they're going to have yellow seeds. If you have big Y, little y, you have yellow seeds. You have, again, big Y, little y here, but you'd have one case where you'd have little y, and you'd have green seeds. And so he was able to calculate, again, this is fundamentally how our genes work. And in this way, he was able to predict for these simple binary one or the other type of outcomes that this is all it takes to explain exactly what it was that he was seeing. And it was only about 100 years later, which is a little bit less than that, about 60 years later, we had figured out that this was in DNA. Turns out, though, it is not that simple, right? So this works for pea plants. This works for very simple attributes where it can be one or the other. But humans aren't short or tall. There's this whole gradient, right? And so that's what I stole from this Wikipedia article, this distribution of human heights. You can't get this from having a tall gene and a small gene. It doesn't work that way. And so in order to get this type of distribution, you need to have multiple genes that all play together that wind up having this variable outcome. And when it comes to something like height, there's also environmental factors that come in, which we're not going to talk about. And so we call this a polygenic inheritance. And we use this to explain things that don't fit this perfect, simple pattern of dominant and recessive. And so that is for normal attributes. Or again, we're not calling a green peed plant a disease state, right? They're still functioning just fine. So, but this is applied, though, to genetic diseases. Well, it actually does. We can use the same principle to apply to genetic diseases, and that some are dominant. You only need one bad copy, and you can show the disease. And obviously, for this audience, the most salient of those is the majority of our Dravet genes are inherited in an autosomal dominant fashion. But it's the hallmark of these are Marfan syndrome, which is a mutation in your connective tissue, and in Huntington disease, which is a rare form of dementia. Much more commonly, what we see genetically is it's recessive. You usually need 2 bad copies in order to have this disease state, where essentially you have 2 copies where this protein is not working, where it's not present, and that's how you wind up with the disease. The hallmark example is sickle cell anemia, but also cystic fibrosis and oculocutaneous albinism, which is just the medical term for being albino, in which case you have 0 copies of functional melanin. And so if I wanted to pick this simply, right, that's a whole lot of words. Some of us are visual learners. And so if we wanted to think about this in a disease state simply, it should be able to say that there's a genetic change, be it dominant, recessive, doesn't matter, that if you have a problem in this gene, that we should be able to predict that you have this certain disease. So if you have a 2 bad copies of your hemoglobin, that we should know that you would have sickle cell anemia. And the inverse should also be true. Everyone that I see that has sickle cell anemia, I should be able to predict that they have a problem, 2 problems to be precise, in their hemoglobin. How would this work for Dravet syndrome? Well, if it was simple, you already know where we're going. If it was simple, we would have everyone with a mutation in SCN1A would wind up looking like Dravet syndrome. Dr. Perry used the term phenotype, and that's exactly what I'm talking about here. That looking at the child, they're having hemiclonic seizures or having prolonged seizures, all those hallmarks that we docs used to recognize as Dravet syndrome. And the inverse should also be true, that everyone I see with Dravet syndrome should predictably have an SCN1A mutation. Again, I think this audience, and if you've been following along, already knows it's not that simple. And this is actually a simplified version of the story here, which is the sad part. So individuals that have SCN1A mutations can have multiple different things. Now, they can have classical Dravet syndrome. They can wind up with genetic epilepsy with febrile seizures plus. They could wind up with febrile seizures only. And I don't even put it here, but they could also wind up with 0 seizures. And they can also wind up with conditions that are abnormal but are not seizure related, like familial hemiplegic migraine. And we're still not there in figuring out what's different about these mutations. Why is it that in some kids this winds up with Dravet syndrome, but for some they're just fine? I have a patient where they have not necessarily Dravet syndrome, but they have a severe form of epilepsy and an SCN1A mutation. Their father has the mutation, but has 0 seizures. They have a sibling with febrile seizures, and they have a sibling with mild epilepsy. They all have the same mutation, but they all look very, very different. So what's different? We also struggle with the other way around. And again, Dr. Perry already stole my thunder a little bit on this one, but it bears repeating in saying that for Dravet syndrome, yes, the majority do wind up having an SCN1A mutation, but it's also PCDH19. It's also this litany of other genes. And there's also conditions where there's no mutations we can even identify. And so, but they all look the same. They all, at the end of the day, Dr. Perry, again, stole some of my thunder, that PCDH19 does look distinguishably different if you're quite experienced in epilepsy. But the point is they look strikingly similar. And so, again, I know you guys just saw this picture, but it's so good that you're probably going to be seeing it a lot today. It's one of my favorite slides that I use in my lectures. And so, conceptually, what's the easiest way to look at this? But it's this picture. And so what you have is this Dravet syndrome phenotype. Again, by phenotype, I just mean looking at this child, they look like they have Dravet syndrome. It's a clinical diagnosis of Dravet syndrome. And you have this large number of them. The one thing I don't like with this picture is that the edges here are pretty gray. There are some kids where, you know, they're pretty darn close to Dravet. We use Dravet-like, but aren't like your textbook Dravet. And the majority of them in this giant picture of a giant circle of SCN1A, there's a huge overlap here in this Venn diagram, right? The one thing I disagree with, there's a lot of SCN1A that is not Dravet. But the point is that the majority of Dravet syndromes are going to have identifiable SCN1A mutations. And then you're going to have all these other sister genes that, again, can look pretty darn similar, if not identical. So SCN2A encodes a very similar protein, and it can look initially kind of similar, although I think, again, most of us are comfortable in making the distinction, which is why it just kind of touches, but doesn't actually fully overlap. SCN8A has significant overlap, but it's not a very common gene. And SCN1B has some overlap. And I want to touch on SCN9A. You'll notice that all of these, first off, they have the same first 3 letters, right? So they sound like they're pretty similar genes. And so it shouldn't surprise us. Okay, they're probably similar genes. They probably do similar things. So it shouldn't surprise us that they have that they look similar. They can all make Dravet syndrome. Well, they're very different, and I'll talk about that in a little bit. But all of these are the same color because they're all fundamentally sodium channel proteins. And I'll talk about that a little bit more on the next slide. But what's interesting is that SCN9A by itself, if you have a mutation of it, doesn't cause Dravet syndrome. We think that it's playing a significant role in determining if you have an SCN1A mutation, whether or not you end up having Dravet syndrome or something else. So we think of this as might be a modifier gene. Another very weird thing, if you have a problem in your SCN1A gene, and so you're not making enough of that protein, turns out that if you have also a mutation, this is true in mice at least, that if they also have one problem in SCN8A, that it actually rescues them. They actually have far fewer seizures and look more normal if they have a bad copy of SCN1A and a bad copy of SCN8A. But, again, with SCN8A, if you have one bad copy, you look like you have Dravet syndrome. So how is it that you have 2 genes where each of them, if you have a problem in them, will make you have Dravet syndrome, but if you have both of these problems, you look more normal? Again, this is what keeps me up at night. Then you have these other genes that are kind of similar in premise as to what they do, but are fundamentally actually the opposite, if we're being honest. So they're also ion channels, HCN1 and KCNA2. These are potassium channels, and I'll talk about a little more in the next slide. Just the important things are a different color here, and that's kind of a hint to say that they do something different. Then you have Gaba receptors, which are a completely different type of ion channel, and a different color. And then you have, I'll put these two in the same color. The one thing I have to disagree here, PCDH19 and STXBP1 do something similar. They're both extracellular matrix proteins, so they should be in the same color. But CHD2 is a completely different gene, which we'll talk about right now. So this is worth harping on because this is really, again, the fundamentals, and it's important to have these fundamentals down pat. So forgive me for repeating. Now, what this slide is illustrating is exactly what I had just talked about, that you have in your DNA 2 good copies of SCN1A, okay? These 2 copies in your DNA both encode mRNA, and this mRNA winds up getting turned into sodium channels. And this is NAV1.1, again, as Dr. Perry had just talked about. Now, the function of these sodium channels is they sit on the plasma membrane of your neurons, and what they do is, in response to voltage, they're voltage-gated sodium channels. In response to a voltage change, they open up and they let sodium ions in. And when those sodium ions come in, they set off what's called an action potential, which is fundamentally the way that neurons speak. Now, in Dravet syndrome, by contrast, again, 80% of children with Dravet syndrome, they have 1 bad copy of SCN1A. They both encode mRNA, but the 1 bad copy of mRNA does not turn into a functional NAV1.1. They do not get that functional sodium channel. So children with Dravet syndrome will still have some healthy functioning sodium channels, some NAV1.1, but they will have fewer. And so again, as I talked about, so this is true for sodium channels for multiple of them, right? We talked about SCN8A, we talked about SCN1B, we talked about SCN9A being a modifier, talked about the weird relationship ACN8, blah, blah, blah. Potassium channels actually do something very fundamentally different. So a potassium channel, it's all the same, except at the end, this isn't sodium. It would actually be potassium that we would put inside the cell, and potassium leaves in response to, again, voltage. It's a voltage-gated potassium channel. When potassium leaves the cell, it actually turns off an action potential. It slows down. It's essentially the internal brakes of these neurons. Chloride, similarly, so these chloride channels, they're not voltage-gated, they're ligand-gated. It's when GABA binds to them, they open up and they let chloride in. And when chloride comes in, it is also like the brakes of the brain. Now, these are fundamentally, yes, they all sound like they're ion channels. They all sound like they do the same thing, but on a cellular level, an biochemical level, they're actually doing often the opposite thing. And yet they all result in Dravet syndrome. Why? How? That's what I would like to know. But that's not all. It gets even more complicated. As I had mentioned, there's also these extracellular matrix proteins. And what do I mean by that? The best illustration I can find of that is for PCDH19. Now, as Dr. Perry had mentioned, this is a distinguishable disease, but it's very, very similar to Dravet syndrome. And what the PCDH19 does is it sits on the outside of these neurons, and what it does is it's looking for other PCDH19. And when it does that, it kind of anchors them all together. So if you have one quote-unquote bad copy of PCDH19, it lives on your X chromosome, and so boys will only have 1 copy. And if you have a quote-unquote bad copy, it's not a big deal, because it's going to look for those other quote-unquote bad copies, and they're all going to bind together no problem. If you have 2 quote-unquote good copies, again, same thing. So for girls, they're going to have half of them that are going to express the normal kind, and half they'll express, again, in this case, also the normal kind. It's only a problem in girls who have 1 good copy and 1 bad copy. And what winds up happening is that half of your neurons, in this case, would express the good copy, and it will only interact with other neurons that have another good copy. On the other hand, the other population that's only expressing the bad copy would only express the other bad copy. Now, this has nothing to do with ion channels, right? This is completely different. It's referring to how these neurons interact with one another. And yet, again, it results in Dravet syndrome. STXBP1 also is kind of in the same premise, but it doesn't have the fancy X-linked component to it. But wait, it gets even more complicated. So I had mentioned CHD2 can also cause what looks exactly like Dravet syndrome. And what does CHD2 do? Well, it's responsible for turning on and turning off genes. What it does is it helps open up the DNA. It's often kind of tightly spooled on these proteins called histones. And CHD2 helps unspool it so that we can actually your body can read the DNA. Has nothing to do with ion channels, has nothing to do with the extracellular proteins, and yet it looks exactly like Dravet syndrome. And so that gets me, again, what keeps me up at night, why do these very fundamentally different things result in the exact same disease? And how is it that you can also, on top of this, have the same mutation in SCN1A, and in both cases wind up with very different diseases? Great questions. I'm not going to be able to answer them today. I'm going to spoil the surprise there. And so now getting to the practical component, and of course I saved myself the least amount of time for this point. As Dr. Perry already mentioned, the vast majority of Dravet syndrome is caused by SCN1A. And it turns out that all these complicated things that I'm talking about are true for actually most genetic forms of epilepsy at this point. And Dravet syndrome, believe it or not, is the most straightforward of all the ones that we're dealing with at this point. And so it is the model. It is actually the one that we're most excited to work with because it's the clearest in a very, very complicated field. PCDH19 is the second most common. Again, it's only if you lump it together with Dravet syndrome, because nowadays we kind of lump it now as a separate condition. And as I had mentioned, it's almost exclusively to girls. And as I had mentioned, we're starting to understand it. There's some recent papers that were published that have talked about some very, very subtle nuances between the different types, different genetic causes of epilepsy, different genetic causes, I should say, of Dravet syndrome. But we're still in our infancy there of telling the difference between SCN1A positive and SCN1A negative Dravet syndrome. And so again, practically, I think it's just worth mentioning that, now this is a very loose boundary. It's right around 2015 was the latest I saw that people with Dravet syndrome would just get SCN1A sequence. And they would do it in this old way that we used to do it, where they would actually count every single letter individually. Now, again, it started earlier than 2015, but the transition really is, I don't think I've seen any SCN1A sequencing since 2015. Now we are way, way better with this technology. We don't just do one gene, we do all of them every time. And on top of that, even though there are, we're doing about 100 genes at a time, actually it's about 140 now, we're actually doing better sequencing of SCN1A when we're doing it as part of this family of 140 genes. We're actually sequencing it about 100 times each time. And so the moral of the story is if you were tested before 2015 and didn't get an answer, and your child has Dravet syndrome, it's worth getting tested again because the technology has changed. And that brings us to the most exciting part, which is gene therapy. Where are we going with this? And so the one that is currently the most active form and has just entered its phase 1 trial is the Antisense Oligonucleotide. And you'll understand why I needed to go through genetics 100.1 before so I can explain what they're actually doing here. The premise of antisense oligonucleotides, if you look at this picture over here, the green marker here that says ASO, that's what we're talking about. So we'll again go through our little journey of we have in the red, it's the bad copy. In the orange, it's the good copy. And as I talked about, the red copy, both of these get turned into mRNA, and there's a bad copy of the mRNA and a good copy of the mRNA. We talked before about how the bad copy does not result in functional protein, which you can see at step 4 down the bottom here, and it gets degraded and winds up going away. These antisense oligonucleotides, what they do is they enhance translation of the good copy. So as we had seen previously, we wind up having just fewer of these NaV1.1 channels, of these sodium channels. And what this antisense oligonucleotide technology is trying to do is to give us more bang for our buck, more NaV1.1 for the same amount of mRNA. This has just entered its phase 1 trial. It just started this summer. The data in mice looks fantastic, and I think we're all very, very anxious to see, can we actually get the same positive results in our kids? The other way that we can do gene therapy is that we actually go into the genome and fix it ourselves. We're actually changing the DNA itself. Now, again, for the antisense oligonucleotides, we're not doing that, right? We're just impacting what happens after that DNA gets turned into RNA. And so this is kind of the next stage of what we're thinking, which what they're going to do, a vector is simply a way of putting something into something else. And in this case, we're using a viral vector. We're using a virus to insert DNA into your genome, into the DNA itself. And so what they do is they package a good copy of SCN1A in a virus, an adenovirus. They target through complicated ways that virus to specific cell types, and we're targeting the one that we know that is most impacted. And what that does is then enters the cell and it squirts its DNA into your DNA, into your genome, and then through lots of obstacles that they've had to overcome, but that should result in increased translation. You should get more mRNA and resulting in more NAV 1.1. And I see I'm over time, so I'm just going to have to just stop it there to say that it's probably going to start their clinical trial next year. Who knows with COVID whether or not it's going to happen, but that's our optimism. I'm looking forward to your questions after this, because I could talk about this stuff all day, but I got to leave time for Dr. Worrell, and she's great, so don't want to cut into her time.
Thank you so much, Dr. Smith, for that talk. I've seen questions roll in, so I'm really excited after Dr. Worrell's talk to dive into those with you.